Kidney disease occurs in about 1 in 10 adults in Australia and can often appear suddenly. This sudden failure, however, 90% of the time is symptomless, with pains showing only at the beginning of kidney failure. The common detection method involves a 24-hour urine analysis. A fast diagnostic tool is therefore necessary. Failure of the kidney is always coincident with the presence of albumin in the urine, a compound that is usually filtered by the kidneys. So rapid detection of albumin in urine can provide vital information to emergency health services. Paper-based microfluidic devices can provide a fast analysis of albumin in urine in a cheap, simple to operate and disposable way. They use the capillary effect to transport a small amount of sample fluid through channels without need for external forces. These channels may contain reagents that interact with the sample should an analyte be present. In the case of albumin analysis, reagents exist on the market that could be added to microfluidic channels to create a change in colour should albumin be in the urine.
As the most important component of urine, inorganic salt plays the most important role during the capillary motion is the factor selected. Because of chemical liquid is included in this experiment, gloves, glasses, and masks are necessary in case eyes or skin get injured. Our first and second attempts found thin channels didn't work well, and an optimized dye concentration made the results unclear to measure. Then we attempted some wide symmetrical structures. The mixing process became better, while some designs were hard to operate. The final designs came out after trying several matches of channels and blocks. The main idea of it is easy to test and measure, with the help of specific channel lines to control testing time.
Several samples of the chosen design were tested to evaluate the impact of urine salinity on mixing efficiency. To ensure the fabrication and testing of each sample was consistent, an experimental protocol was followed. The first step of fabrication was to trace out the printed channel template onto coffee filter paper in pen and then crayon. All excess crayon fragments were brushed off to prevent contamination of the channel. Each sample was on a separate piece of paper to prevent dye leaking from one sample to another. The samples were then placed into an oven at 110 degrees Celsius for 3 minutes. For the mixing testing, 2 glasses with 30 milliliters of water were prepared with 10 drops of yellow and blue food dye. The samples were then taped onto a clean, dry and non-porous surface. 2 separate syringes were used to draw 0.5 mils of yellow and blue dyed water to ensure that no contamination occurred. The yellow and blue fluid was carefully dispensed into the left and right reservoirs respectively. A photo of the sample was taken once the fluid had stopped flowing. The same steps were repeated with 2 different concentrations of yellow dyed salt water. 3 trials were completed for each solution. The average grayscale intensity of the reservoirs and the mixed fluid was measured using ImageJ. The average mixing efficiency for each solution was then calculated using the equation shown.
The length for the straight side of the main channel is 15 millimeters. A pixel to millimeter scale is set in ImageJ using the yellow line as a measure in each image. A segmented line is used to draw a connection between the blue and yellow reservoirs. The image is converted to 8-bit grayscale and normalized. Then, the intensity of the pixels along the line are taken and stored. Using the equations previously shown, the average mixing efficiency was plotted against the salt concentration. There is a slight increasing trend between salt concentration and mixing efficiency, with average mixing efficiencies recorded in the range of 50 to 60%. However, one of the trials was a clear outlier. The blue reservoir in this trial was completely mixed in the green, and thus there was no value recorded for the intensity of the blue reservoir. The average mixing efficiency was plotted against the salt concentration with this trial removed. Now, a slight decreasing trend can be seen. It was concluded that there is no significant relationship between salt concentration and mixing efficiency. In many of the trials, the distinction between different hues used in the reservoirs can be easily observed by eye. The design of the device restricted the flow to be laminar near the islands in the center of the main channel, which helped to improve mixing of colors. This paper-based microfluidic device design could be adapted to include more reservoirs for a simultaneous bioassay of a single analyte. For example, a bioassay of urine could be performed on a single device to measure the concentration of albumin, creatinine, and other factors using this device.
Microfluidic devices are important for the detection of albumin in urine as an indicator of kidney failure. This study determined the effectiveness of several patents in terms of mixing efficiency. Having selected one, the relationship between salinity and mixing efficiency was also investigated. It was found that with increased salt levels in the water samples used to represent urine, there was an increase in mixing efficiency across the device.